Good afternoon. No. Good afternoon. So, I'm Jun Takahashi from Kyoto, Japan. Do you know, maybe you know the Surgeon Garden. And this is Dr. Shinya Yamanaka. Do you know why these two guys are aside? Yeah. Correct. No. Yeah, they won the Nobel Prize in 2012 for Nobel Prize for medicine, physiology and medicine. And Shinya Yamanaka has discovered the iPS cells. So iPS cells has two major characteristics. One is iPS cells can proliferate infinitely. And also, iPS cells can give rise to a variety of cells. It is called pluripotency. So iPS cells can give rise to neurons or muscle cells, heart cells, blood cells, cartridges. So we can use iPS cell technology for two main purposes. One is disease to reveal the disease mechanisms or for the drug development. Another one is cell therapy. So iPS cells can made from your cells, your skin cells or your blood cells, and then make neurons or muscles and transplant to you or patients. So this is our clinical trial scheme. So in our institute, it is called SIRA. We get blood cells from the healthy volunteers, and we make iPS cells in very clean room. And we have established the clinical grade iPS cells. And we make the dopaminergic progenitor cells from the iPS cells. And we transplant to the patient. And we evaluate the safety and efficacy. The second question is where is trial taking place? It is Kyoto University Hospital in Kyoto, Japan. I am a neurosurgeon, so I have been working for the functional neurosurgeon, like deep brain stimulation for the Parkinson disease. So originally, I am interested in such kind of new therapy. And this is a picture of the Kyoto. So this is a member of the Kyoto University Hospital who is involved in our clinical trial. So there are so many people are working for this trial. And how many participants will be involved? It is seven patients will be involved. So this is the design of our clinical trials. So this is phase 1 and 2 study and single institute in Kyoto University Hospital. So this is non-randomized and open. And this is the basically so phase 1 study. So there's no control group. We just compare before and after the surgery, and 7 patients. But for the first patients, we Japanese regulatory request us to do the surgery one side and wait for 6 months and confirm the safety and then inject the cells to other side. So, but finally, so we will perform the cell transplantation for the 7 patients. What is involved? Surgery or number of assessments or type of assessments. So we inject the cells by the stereotactic injection. So stereotactic injection of the cells into the bilateral putamen. And after that, we will evaluate the neurological symptoms and brain images. images. So this is an illustration of our surgery. So for this purpose, we have developed a long, thin needle for the cell injection into the brain. And under general anesthesia, we make the burr hole, small hole to the skull. And through this hole, we inject the cells for the bilateral putamen. And this is not autologous transplantation. This is allogenic transplantation. So the cells of others will be injected for the patients. So we will give immune suppressant drugs. It's tacrolimus for 1 year to repress the immune response. So number of the cells will be about 5 million cells per person for both sides. So we inject the cell aggregation, and the average diameter is 400 micrometer, and we took these cell sphere to the needle, and this is the picture of our surgery. So we just inject the cells through 3 tracts to the putamen. And we inject 4 points in each tract. So in total, we inject the 12 points. And 1 point is 200,000 cells. So totally 2.4 million cells per side. Follow-up period is 2 years, and primary endpoint is adverse events, especially for the tumor formation or dyskinesia or hemorrhage. For the second endpoint, we will evaluate the symptoms like UPDRS score or off time period or ERS stage or PDQ-39. Not only the symptoms, we will evaluate the brain images by using PET. We will do the 3 PETs. So one is the F-DOPA. It is for the dopamine synthesis. And also, we will monitor the immune response by using ZE180. Another concern is cell differentiation, cell proliferation. So for that purpose, so we will use the FLT PET to detect if the cells are dividing actively. And also we perform that scan to evaluate the dopamine transporter. And how are you stratifying the patients' characteristics of the ideal participant? So this is the summary of our criteria. So one is uncontrollable by medical treatment, and years is 50 to 69, and suffering from Parkinson's disease more than 5 years. And scale is stage 1, 2, 3 in on stage, and 3 to 5 at off stage. So like this stage is the target. And also important thing is the patient should be responsive for L-DOPA, because we graft the dopamine producing cells. So if the patient is too severe and not responsive for dopamine, so it is useless. So the patient have to be responsive for L-DOPA. That's the important point. So this is the day of surgery. So before surgery, we evaluate at least 6 months, and after that 3 months, 6 months, 1 year, and 18 months and 2 years. So at this point, we evaluate the symptoms or brain images. Okay. So when will it start and finish? As I mentioned, we started our clinical trials in 2018. Maybe, so we will finish it 2022. And we will get the final outcome in 22 or 2023. Okay, this is the final question. Okay, and so we have been working for the Parkinson's disease about 20 years. So a lot of members inside or outside the Kyoto University have involved. Okay, thank you very much for your attention.